This is Maria Talara for my microbiology lab experiment, biochemical testing for microbial identification using carbohydrate fermentation. For this experiment, I have set up my area to do my work. So first, we wash our hands to make sure for infection control, and then we gather the supplies that are asked from us. So we do have, of course, our gloves for personal protective equipment. Then we have our two cultures, the S. cerevisiae and S. epidermis. And then we do have our three kind of carbohydrates we're gonna use, which is the fructose, glucose, and mannitol. Mannitol is an alcohol sugar, and glucose is often used when the plant do photosynthesis or the breakdown of glycogen. And then fructose is usually found in fruits and vegetables. And then we do have our lighter and our candle that we use to sterilize the lid of our cultures, so we don't have infection or cross-contamination of our specimens. And then we have the Durham tubes, which is soaked in alcohol. We'll take it out and dry it out later, so we disinfect them and not cross-contaminate our specimen, so there's no introduction of another microbe. And then we have the two pipettes. We will need to pipette our culture to our test tubes. And of course, you use alcohol to sterilize the tool also, so we do not have introduction of a new microbe. Then we have our phenol red test tubes in our test tube rack, which is easy to which is actually good for us to keep it upright. So what I learned about the phenol red is that it is red because it's a pH indicator. So if fermentation occurs, the color changes to yellow because of all the the end products, which is like the lactic acid and all that alcohol and all that stuff that makes the solution acidic. And then also the Durham tubes are gonna be used to catch any gas that will come from the fermentation process, which is usually the gas production. And that's our supplies. And usually we open this and drop half of this. Oh wait, so for your test tubes, we need to make a marker and pair each tube. So this will be for glucose, but one S. epidermis and S. cerevisiae, and same goes to these tubes, and same goes to this tube. So this row will be your S. epidermis, and the other one is the S. cerevisiae. And then you drop the sugars for each. So this one will be the mannitol, half of the powder goes to the other tube and goes to the other one, so on and so forth. Make sure do not confuse them. Mark your test tubes and use the right powder. And then we do drop also our Durham tubes to the test tubes, making sure the opening of the Durham tubes is facing the bottom of the test tube to collect the gas that might occur during the fermentation. And then I think then we drop our culture to the sample, and that making sure we sterile all the equipments before dropping the culture, and then we culture it for at least, I would say, 24 hours to get more of the reaction, and then we'll review the results. Thank you.